So the topic of this morning's session is the publication of research involving the H5N1 avian influenza adaptation to mammalian hosts. And we have a great panel here with very different perspectives on this problem that we're going to get a chance to hear from. So this research that we're going to be talking about today was performed at Erasmus University under the direction of Dr. Ron Fauci, who is one of our panelists, who will be our first speaker. This research was performed and then written up and submitted to Science magazine for publication. That Science manuscript, of course, went through the hands of the editor-in-chief, Dr. Bruce Alberts, who's also one of our panelists this morning and will be presenting the perspectives from the Science magazine. In the process of doing the research and in preparing the manuscript for publication, it was identified as being potentially dual-use research of concern, or what we like to call DIRC. And so as being identified by the U.S. government as being dual-use research of concern, it was sent to the National Science Advisory Board for review and recommendations. Now, it was the U.S. government then that tasked the National Science Advisory Board to review this, and representing the U.S. government and the funding agency for this research is Dr. Tony Fauci, the director of the National Institute of Allergy and Infectious Diseases, again, one of our panelists. Very pleased to have him here as well. So after the paper, at a nearly identical timeline to what was happening with Dr. Fauci's research and Science magazine, a second group at the University of Wisconsin, headed by Yoshi Kawaoka, was doing similar types of research in adapting the H5N1 virus to mammals. And this research was written up and submitted to Nature magazine. And likewise, this research was funded by the National Institutes of Health, and in the process of the research and in the publication, it was identified as potentially, again, dual-use research of concern. And this paper was also forwarded simultaneously with the Fauci paper to the National Science Advisory Board for Biosecurity for review. So when it got there, it was reviewed, and the NSABB made recommendations. And representing the NSABB today is Dr. Mike Osterholm, who's a member of the NSABB since 2004. And likewise, I'm the acting chair of that board at this time. So the two of us represent then the National Science Advisory Board for Biosecurity. So the recommendations that the NSABB came back with was that these papers would in fact not be published in full. We recommended to the U.S. government that the papers be modified, the results and experimental details be redacted, so that somebody with malevolent intent could not exactly replicate the results. And that was a recommendation that went back to the U.S. government. The U.S. government then accepted those recommendations and went back to the journals and to the investigators. So today we have a lot to talk about, but one of the things we can't talk about are the actual details of these papers. The details are still confidential, as per the NSABB recommendation, and so we won't get down to that level of detail today, but I assure you there are many things to talk about because of these recommendations. I've tasked the panelists to first try to communicate to you exactly what has occurred and then what the implications and future are in this area. Each of them have a very unique perspective on what's on this work, and will obviously not always agree. We'll try to get through these talks in time to have about 20 minutes of question and answer, and so scribble down your questions and hop to the microphone fast after the last speaker. So with that, I'd like to invite Dr. Fauci to the podium.
Thank you, Paul. I'd like to thank the organizers for allowing me to speak here. In the next 10 minutes, I will run through the experiments globally, what we have done, why we have done it, and what the context of these experiments was. And also, I take some time to get rid of some of the misperceptions that have come through the lay press about our work. This work was done by a large team of investigators in my laboratory. In the first place, Sander Harris, a postdoc in my laboratory. So this work really got started back in 1997 when there was the first incidence of human exposure to fully avian, highly pathogenic H5N1 virus, including the first human deaths related to this virus. And it was very quickly recognized that these viruses were coming out of poultry. There were poultry outbreaks in Hong Kong. The Hong Kong government started to kill all the poultry in Hong Kong, and by that time, after that, it was thought that this outbreak was over, but that appeared not to be the case. The virus remained enzootic in Southeast Asia, and from 2004 onwards started spreading over numerous countries on 3 continents, including parts of Asia, Africa, the Middle East, and Europe. In many of the countries where the virus landed, culling was not the first option. And as a consequence, these outbreaks are still going on today. These viruses have caused hundreds and hundreds of millions of poultry deaths, and on occasions these viruses have also been transmitted to various mammals, including cats, dogs, pigs, tigers, and also, importantly, they have spread to humans. And from 1997 onwards, the question has been, in the influenza community as well as in the public health community on the whole, can this virus go airborne, and can this virus ever trigger a pandemic? As it usually goes in this type of, when this type of questions arise, the research agenda is set under the umbrella of a variety of organizations, such as NIH, FAO, OIE, WHO, who are putting this question, can H5N1 go airborne, very high on the research agenda. And as a result, various calls for proposals for research were then drafted by the EU, by the NIH, and by many other organizations. And scientists then, of course, start addressing these questions. And here's a list of 5 or 6 publications already that have come out since 2006 where scientists have tried to address this issue for H5N1 virus, but also for other animal influenza viruses that are circulating in the reservoir. Can these viruses go airborne? At Erasmus, we followed these events here on the slide. In 2005, there's been a call for proposals from the NIH to start the Centers of Excellence in Influenza Research and Surveillance Program. There was a broad agency announcement in 2005 that we responded to together with scientists at Mount Sinai School of Medicine. Our proposals were reviewed favorably, and our contract was rewarded in 2006. At that time, we also started to build our special facilities for this type of research. We do not take this lightly. We purposely built ABSL3+ facilities specifically for working with airborne pathogens. We also obtained an explicit permit to work with these viruses from the Dutch government. We applied for such permit from the Ministry of Infrastructure and the Environment, and the Ministry was advised by the Dutch Committee on Genetic Modification, and both of these organizations, so the Committee for Genetic Modification and the Ministry, judged that under the conditions that we created in the ABSL3+, we could do this type of research with negligible risk to humans and the environment. In 2007, our facilities were cleared by the US CDC as well as our local biosafety office, and this clearance by the US CDC was necessary because overseas laboratories that are funded by NIH need to get approval to work with select agents. And since 2007, we've also been, of course, inspected by the Dutch ministries. The work then started in 2007, and since 2007, we have reported our progress through semi-annual reports to the NIH, but also through the normal routes of publications and conference presentations. But it's important to note that until the summer of 2007, no progress could be reported about transmission because the H5N1 viruses until that time did not transmit, neither in our studies nor in studies by CDC and other laboratories. So what do? The experimental setup is very simple. We infect a ferret in a cage with an H5N1 virus, and we then simply wait whether this ferret will sneeze or cough or breathe, such that the virus would end up in the ferret in the cage next door. And these ferrets are separated by 2 steel grids that prevent direct contact between the animals, but still allow airflow between the animals to occur freely. These animals are not in an open facility. They are housed in a BSL-3 facility, low-pressure HEPA-filtered room. And in this room, all the experiments are carried out in Class 3 biosafety cabinets and Class 3 isolator units when we do animal experiments. And so in many ways, this is similar to the conditions in a BSL-4 lab, except that in a BSL-4 lab, most times the investigators are packed in spacesuits, whereas in our case, we actually wrap our experiments in these spacesuits. So what did we do? We took an H5N1 virus, and we took a mutated H5N1 virus, and I cannot give you more details about what those mutants are, and we passage them in ferrets. And then we see whether those viruses become transmissible by aerosol. And we find that the H5N1 wild type does not transmit, so it replicates in the donor ferrets, but not in the recipients. But if we do this with a mutated H5N1 virus, we can see that in 3 out of 4 cases, the virus is transmitted via aerosol or respiratory droplets. We then take this virus from this ferret, from one of these ferrets here, and try to do it again. And indeed, we can now, in 2 out of 2 ferrets, see transmission again. So this is the first time we have worked with an aerosol-transmitted H5N1 virus. That on itself is an important finding, but on top of that, we now know exactly which mutations and which biological traits are necessary for H5N1 bird flu virus to become aerosol transmissible between ferrets. Now these are the main conclusions of our work, and because our paper still hasn't been published, but the press has picked up on our story, there's a lot of misperceptions what you can and cannot conclude from these studies. Now, the first thing that we've heard many times in the press is that this virus would spread like wildfire if it would come out of our facility. But we do not think that this is the case. So we have shown aerosol transmission, but the efficiency cannot be deduced from our experiment. This is not a quantitative setup. But even if we try to extrapolate a little bit further, if we look here, this is pandemic H1N1 from 2009, and what you can see here is in the lines is the shedding of virus from animals that have been inoculated, and then the bars are titers of virus at different days after infection that have received the virus by aerosol. And we can see for pandemic H1N1 in 4 out of 4 animals, we can see massive virus replication, and this replication starts as early as day 1 in the recipient ferret, and generally all ferrets by day 2 are starting to shed viruses at high amounts. But if we compare this to H5N1, we can see that first of all, it doesn't transmit in 100% of the cases. Secondly, the virus titers that are being shed in the aerosol contact animals are much lower. And thirdly, a lot of the animals actually do not start shedding virus until day 3, 4, or 5. And so, remember, this is not a quantitative model, but if one would deduce anything from this, the efficiency of spreading, we would have to conclude that this virus does not spread yet like a pandemic or a seasonal influenza virus. The second misconception is that the virus would be highly lethal if this would ever come out. But also here there is some facts to explain. It is absolutely clear that H5N1 is a highly pathogenic virus for chickens. You inoculate a chicken, the chicken will drop dead. But in mammals, this is certainly not the case. In mammals, it is known that if we infect ferrets, for instance, with very, very high doses, 10 to the 6th of virus, and put it straight down into the lungs of the animals, yes, the animals will drop dead, and they will do so by around day 3. But if we inoculate ferrets or other mammals via an intranasal route, the animals generally do not get sick at all. They might get a little bit of flu, but they certainly do not drop dead. Now, the virus that we have used does cause disease when we put it via aerosol, when we put the virus in the nose at very, very high titers again, right? 10 to the 6 in the nose, we get some neurological symptoms, and it's known that this is very typical for the virus we use, but not a general observation for H5N1. But it's important to note that the mutant virus only causes disease in 1 of 8 animals if we inoculate at this very high dose. But if we now look after aerosol transmission, we actually see no disease, no severe disease at all in any of the 7 animals that received virus via aerosol. So this virus, although it is highly lethal to chickens and highly lethal if you put it directly down at high titers in the lower respiratory tract, it is certainly not highly lethal if ferrets start coughing and sneezing to one another. It's also important to note, and I'm sorry that my slides have shifted going from Mac to Windows here, that when ferrets are pre-exposed to seasonal flu, they are fully protected from developing severe disease. So if we compare that to humans, and you all have been infected previously with seasonal flu, it would be unlikely that you would have no cross-protection against a virus like H5N1. And so very few individuals would actually develop severe disease, but most of them would actually be protected by cross-protective immunity. So I'm going to conclude that if you ask me, should this work be done, the answer is yes, but we have to assure appropriate biosafety and biosecurity conditions, and I think that we have met those biosafety and biosecurity conditions very well. And why we would be doing it was, first of all, of course, in the interest of fundamental science. This is the first time ever that we have made an animal flu virus transmissible via aerosol, and this will really allow us to better understand how viruses are becoming aerosol transmissible, and therefore will aid in our understanding for pandemics of the future. Secondly, we think that there's a direct implication for public health. First of all, we try to prevent potential pandemics by putting out warning systems to the countries where H5N1 is circulating. And until today, or until a couple of months ago, the expert advice to those countries about the public health risks of H5N1 were not unanimous. And now they are going to be more unanimous now that we've shown that H5N1 can become airborne. Secondly, because we now know the biological traits of these viruses and the mutations that are associated with those biological traits, we might be able to do better surveillance. We might be able to study viruses in outbreaks and to see whether some of these mutations are already occurring. And indeed, we see over recent years that the mutations that we find and that Yoshikawa Oka found are becoming more prevalent in the field, and that is not good news. If we fail to prevent a pandemic, then we might still be able to handle it better when it hits. And we can do so by testing vaccines, antivirals, clinical management, better with viruses that have the proper biological traits, viruses that are transmitted via aerosol. Now, you've all noticed that there has been an enormous discussion in the public health field, in the lay press, and because there was so much controversy, we and Yoshikawa Oka's group and all other groups that did this type of work or had the capability of doing this type of work called for a moratorium until the end of March. And the reason to do this was to give more room for the discussions as we are doing here today, hopefully. There are clear challenges and there are clear opportunities from our research, and those need to be handled as well as possible without getting into the wrong conclusions by acting too quickly. And it is important, I think, that everybody who is making decisions about this work know the details of our study. Right now, there are too many misperceptions about this work simply because the work cannot be evaluated by everybody involved in making the judgments, and therefore I think it's important to publish this work in detail. I'll stop there.
Thanks, Rod. So our next speaker is Mike Osterholm, and he's going to present the view from the National Science Advisory Board for Biosecurity. Mike.
Thank you, Paul, and thank you, Ron, for that very thoughtful presentation. Tony, Bruce, Ron, Paul. Today I'm here to present a very, might say, specific focus on this issue. I'm not here to talk about whether this research should have or needs to be done. I think we all recognize H5N1 is a very important potential pathogen for humans. I'm not here to talk about what the future research agenda should look like. I'm here to talk about a specific process that took place with the National Science Advisory Board for Biosecurity. It began in mid-October. For many of you, you may not even be familiar fully with what the National Science Advisory Board for Biosecurity is. It's a committee of 23 subject matter experts who were empowered in late 2004 to provide advice to the federal government. Of the 23 subject matter experts, 15 have a primary interest or expertise in infectious diseases. It includes the incoming president of ASM, the incoming president of the IDSA, and a number of other major public health and infectious disease experts. There are 15 ex officio members, including Dr. Fauci and others from the federal government. Our single task as charged is to advise the U.S. government how to minimize the risk of that information, products, and technology from life sciences research could be misused to cause harm to public health and safety, agriculture, plants, animals, the environment, or material. We do not, as a routine, review anyone's research. We have largely been a body providing advice. You can find more about the NSABB in our reports at www.biosecurityboard.gov. The issue that came before us and was brought to us by the U.S. government and with which we were charged with reviewing was 2 U.S. government-funded studies on respiratory transmission of H5N1 as submitted to publications which you've just heard about. The manuscripts raise the dual-use research concerns in that they contain information that could be utilized to create a potentially human-transmissible form of H5N1 that could be intentionally released to threaten public health and security. In recognition of this, the U.S. government tasked our board with reviewing the unpublished manuscripts and making recommendations on their communication. All of our formal communication has gone directly to the U.S. government. A working group was established when this task first came to us, and the purpose of this was to assess the dual-use research implications of the 2 unpublished manuscripts and to consider both the risks and benefits of communicating the research results, and to provide a draft findings and recommendations to the full NSABB regarding the responsible communication of this research. The NSABB working group included a subset of NSABB voting members. It included U.S. federal agency representatives. In addition, outside subject matter experts were brought in to advise the group. The deliberations included in-depth review and discussion of the manuscripts. To say that this was in-depth would not do it justice. Over a period of not quite 2 months, there were many hundreds of hours of conference calls scheduled with this group in terms of going through in detail. We also had a question and answer session with the PIs themselves for the 2 manuscripts. We also utilized a previously created NSABB communication tool as a framework for the risk-benefit analysis communicating the information in the manuscripts. To date, the NSABB has largely spent its time understanding how we can best communicate research and believing that the full communication of research was by far the highest priority, as that is what is going to be the ultimate way that we really address many of the emerging infectious disease issues of our time. We developed draft recommendations for the full NSABB consideration. In the NSABB deliberations, which were in closed sessions due to the discussion of the unpublished information, we issued our findings and recommendation to the U.S. government. I must tell you that this group is a very intellectually advanced group as a whole, with 1 or 2 members stepping aside here. This is a group that is like trying to herd tigers. You do not make this group come to a singular conclusion because you want them to. Yet the vote for our recommendations were unanimous, 23-0. So I just want to emphasize that that was by itself, I think, an important statement. Points to consider here in terms of our communication tool in assessing the risk and benefits and communicating this research information with dual-use potential was first a general overall overview of the research information with dual-use potential, and we did that. We looked at the risk analysis, what are the potential issues there in depth. We looked at the benefit analysis in depth, and then we did a risk-benefit assessment. It was based on that combination of deliberations that we then formulated our recommendations. I don't have time in the very limited period we have here today to go through in detail this risk-benefit analysis, nor can I go through it fully because of the confidential nature of the materials. But trust me, we looked very carefully at both sides of this issue. In terms of our recommendations, we unanimously recommended that neither manuscripts be published with complete data and experimental details. We further recommended that the conclusions of the manuscripts be published, but without experimental details and mutation data that would enable replication of the experiments. I think it's fair to say that these are probably the 2 most famous unpublished manuscripts in modern life science history. Text to be added describing the goals of the research, the potential benefits to public health, the risk assessment performed prior to research initiation, the ongoing biosafety oversight, containment, and occupational health measures, the biosecurity practices and adherence to select agent regulation, and that addressing biosafety, biosecurity, and occupational health is part of the responsible conduct of all life science research. I think you just heard from Ron a fair amount of that presented today, which is very helpful. The board also recommended the NSABB develop a statement that explains their review process and the rationale for the recommendations. This statement will be provided to journals to consider for publication, which I'll show you in a moment has been done. We also encourage the authors to submit a special communication commentary letter to the journals regarding the dual-use research issue. The last thing that we wanted to do was stop communication. We wanted to enhance communication, but communication for which we could one day basically say we hadn't gone too far and need to redact or retract. Our rationale is if the influenza A H5N1 virus acquires a capacity for human-to-human transmission, the world could face a pandemic of significant proportions. Also, before these experiments were done, it was uncertain whether avian influenza H5N1 could ever acquire the capacity for mammal-to-mammal transmission. Now that this information is known, society can take steps globally to prepare for when nature might generate such a virus spontaneously. We, in the first instance, understood and appreciated that the real issue here was a Mother Nature-made influenza pandemic, and we must not do anything to detract our ability to better prepare for that. At the same time, these scientific results also represent a grave concern for global biosecurity, biosafety, and public health. Could this knowledge in the hands of malevolent individual, organizations, or governments allow construction of a genetically altered influenza virus capable of causing a pandemic with a mortality exceeding that of the 1918 Spanish flu epidemic? Let me make it very clear right now, because this has been an issue of great diversion. The case fatality rate, as currently determined for H5N1, whether it's an order of magnitude off or not, whatever it is, was never a major consideration for our deliberations, because we recognized even if this particular virus were to actually have a 20-fold reduction in mortality from what's witnessed today, it would still far exceed that of the 1918 pandemic. Unfortunately, far too much has been made of that issue, and it is a diversion from what I believe are really the important issues at hand. The NSABB found the potential risk of public harm to be unusually high magnitude. In formulating the recommendations, NSABB tried to balance the great risk against the benefits that could come from making the details of this research known. Because the NSABB found that there was significant potential for harm in fully publishing these results, that the harm exceeded the benefits of publication, the board therefore recommended that the work not be fully communicated in open forum. Now, we did share both in Science and in Nature our statement, and that is beyond what we formally presented to the U.S. government. Let me just really emphasize an important point here. We believe that our role, as charged by the U.S. government, was to give advice on the dual-use research issue here. But in our statements, we made it very clear that this had to be the beginning, not the end of the discussion, that it must include a global life sciences input, that in fact what we wanted to do was do no harm, but that did not mean that ultimately down the road with further discussion and further deliberation that there might not be a review of this that would allow us to say we might make in fact, more information available. So, in fact, the NSABB never saw itself as the court of final vote. It saw itself really as the court of the first vote, that really to cause a pause for us to think about this carefully. We've tried to provide additional clarification on our role. Paul authored a paper here in Nature, a Q&A section with input from a number of the members here. I would urge you to look at this. He also provided a rationale for the recommendations, impact and implications in MBio, as well as Rob Webster, one of the expert consultants to the NSABB, published a similar type of commentary in MBio looking at this issue, and Rob's input into this process was very important. Some of us have tried to also deal with the ancillary issues, case fatality rate, the real potential for countermeasure production and availability, and those articles are also available. Let me just conclude with the fact that we had input from a number of people, and a fair amount of it was unsolicited but very helpful. This is a message we received from an influenza virologist here in this country. Paul is aware of who this came from as it was shared with the NSABB. I just want to share with you this note, which was not that uncommon in what we heard. I'm an influenza virologist myself, and we routinely create viral mutants in my lab using reverse genetics, so I have a good sense of the technical issues involved. As such, I can recognize that some outspoken researchers in our field have been underrepresenting the increased risk that would be entailed by full publication of the specific mutations versus the current situation, where only the general outline of the ferret passage scheme is known. A ferret passage experiment is expensive and technically demanding, and could only be done by a handful of labs in the world. Once the mutations are public, individuals in my lab or many other labs could generate the mutants in a few weeks, given several $1,000s for gene synthesis. He went on to say, I remain agnostic about what is the best policy going forward. I recognize that there's also important potential benefits from this research and think that the research along these lines does have valid scientific and public health justifications. But these benefits need to be carefully weighed against the real risk, and I am definitely concerned that there has been a rush to judgment for full publication within our own research community. We heard that message loud and clear from a number of influenza researchers. And so it is, we're urging that there is not a rush to judgment, that in fact we really spend the time and the effort in careful deliberations, and that's what we believe the NSABB has started that process. Thank you.
Thanks, Mike. We're going to turn the podium over to Dr. Tony Fauci now. Tony.
Thank you very much, Paul. It's a pleasure to be here. I'm going to go as quickly as possible through this because I really feel we need to get some chance to have some discussion. So I'm going to focus fundamentally on the government response to the recommendations and the subsequent events, but I also want to give a really brief background about why we feel at the NIH, why these studies are important. It really goes back several years when we first became really quite aware of the threat of pandemic influenza. This was a government-wide involving CDC, FDA, the department, and the NIH and other agencies to develop a strategic pandemic influenza preparedness plan. As you see here, it involves multiple components from surveillance through research, including vaccines, antivirals, communications, etc. At the NIH, as many of you know, our research agendas with influenza include a variety of issues going from fundamental basic research to clinical research and focusing on the development of therapeutics, vaccines, and diagnostics. This has been accompanied by a very robust increase in the resources allocated to influenza. As you see here, we went from a relatively small amount, and then when you had the real acceleration of interest in the H5N1, particularly in Southeast Asia and Northern Africa, we had a major increase in our influenza. This flatness of the curve just reflects everything that's gone of the NIH over the last several years. With that, the influenza basic research, we'll put aside clinical trials, we'll put aside therapeutics, and look at what we do for basic research. As you can see, it involves issues from viral evolution, immune response, pathogenesis, molecular analysis, but particularly and importantly, the work that you just heard from Dr. Fauci, namely host adaptation and transmissibility, which is actually the aim of the studies that were done. And as Dr. Fauci very correctly said, this was work that we were interested in pursuing and felt were very, very important in our ability to address the issue of pandemic influenza in general and specifically H5N1 as a threat. Now, let me give a minute or two on what the NIH, NIAID research review and oversight is. It's a dynamic process with multiple steps. And again, I'm just going to try and give you the message and not go through every single word here. First, there's a research agenda. There's internal portfolio reviews. We get input from outside experts, blue ribbon committees that are called not infrequently, and we get progress reports, as Dr. Fauci mentioned in his report. The individual projects are peer-reviewed by external and internal subject matter experts. We have institutional biosafety committees we call IBCs. We have progress reports from the PIs annually to the NIAID and intermittently to the program offices. There is select agent oversight. Namely, if you're working with a select agent, you have to be pre-approved in training and facility. If it's a select agent with an animal, it's the USDA. If it's the select agent with a human, it's the CDC. They do site visits, security checks, biosafety, biosecurity, and training compliance. There's oversight at the peer review level and evaluation for some contracts, such as the Centers for Excellence in Influenza Research and Surveillance, which is the overall body which subcontracted to Dr. Fauci to do his work. And then, as you heard from Mike, we have an NSABB consultation. The studies in question, I don't need to go over them. They were very nicely described. So the recommendations, again, I won't go over just what, my feel for them. They're there, they're in print, you can read them yourself. And it was, as Mike said, to restrict communications of these particular results for the reasons that Mike described very clearly. and also the need for a rapid and broad international discussion, and actually even discussing the possibility of a scientific community participating in a moratorium. How did the NIH respond to the NSABB recommendations and subsequent events? First, as you heard, we accepted the NSABB recommendations, delivered the recommendations to the authors and to the journals. You'll hear from Bruce Alberts from Science. We worked with the journals regarding the implementation of what would be a redacted versus unredacted manuscript, namely publish it as suggested by the NSABB in a restricted form that doesn't give the methodology or the data, but have the opportunity in some way to get the full manuscript for those who have a need to know from a scientific or public health standpoint. The investigators voluntarily had a voluntary pause, a moratorium. We worked with them. We encouraged them. They did it voluntarily. We at the NIH and CDC imposed a pause on our intramural scientists. What was the reason for this? The reason was that we had a lot of noise in the system. As many of, if not all of you know, there was a lot of going back and forth in the press. There was more said in the press than there was at the scientific level. And we felt that we needed to call a timeout, take a look at what's going on, and reevaluate the process. As part of that, I myself personally, as were my colleagues in the government, very concerned that this was perceived as a USA-centric process and a U.S. government-centric process. And for that reason, we recommended and supported the international meeting which took place on February 16th and 17th at the WHO in Geneva. Now, the consultation of the WHO, which was really a consensus body to make recommendations with no authority, and I'll very briefly go over that. Of note, it was a 2-day meeting in which both investigators had the opportunity to present in a much longer forum than we have here, and a lot of time for questions and interaction. Based on that, what we did, what the consensus, which was led by by WHO and Keiji Fukuda, was a reaffirmation of the Pandemic Influenza Preparedness Agreement. Namely, make sure when you do research, you involve and show the courtesy and respect for the countries from which you get the specimens. There was new data on 2 manuscripts were presented. The original data was represented, and there was a substantial clarification of the data in both manuscripts, particularly one manuscript, namely the one that you heard about from Ron just a few moments ago. There was a recommendation to extend the moratorium that was already a voluntary 60-day moratorium. We were almost 30 days into it at the time. This is important. The recommendation was to ultimately publish the manuscript in full with certain conditions, namely underscoring the emphasis on safety and biosecurity and enhanced communication of this balance of the risk-benefit ratio. They expressed the futility of a redacted versus an unredacted approach to the published manuscripts. They recommended the investigators provide new data and clarification in revised manuscripts to the NSABB, and to hold larger international meeting with broader representation. This was a meeting that was very heavily weighted with international influenza experts and influenza public health people, representation from the journals, representation from NSABB with Paul and myself, Nancy Cox, as representation of the U.S. federal government. The response of NIH now to the broad picture, including NSABB and the meeting in Geneva, so it was to continue to support the NSABB recommendations regarding the original manuscripts, to support a revision of the manuscripts to include new data and elicit clarifications of old data, to reconvene the NSABB to re-examine the revised manuscripts, and to endorse the WHO consensus for the extension of pause on the conduct of H5N1 transmissibility research and fortification of the biosafety and biosecurity issues. And also to conduct an internal inventory of our intramural research activities involving dual-use research of concern, to conduct an inventory of existing and pending extramural grants and contracts involving the dual-use research of concern, and to develop an interim process for enhancing the evaluation and the monitoring and the communication of DIRC, namely dual-use research of concern at NIH, and to participate in an interagency process to develop interim and long-term policy on dual-use research. Thank you.
Thanks, Tony. Great talk. So our final speaker then is Bruce Alberts, Editor-in-Chief of Science. Bruce.
Thank you. It's a pleasure to be here today. I'm going to give you a little background. Before I came to Science Magazine, I was president of the U.S. National Academy of Sciences, and I was involved in these issues early on. In fact, right after 9/11, it was recognized that the kind of issue we're dealing with was going to come up in the future in biology. Of course, the physicists have been dealing with these kinds of issues for a long time. And a special committee of distinguished scientists plus security experts was established at the national academies, and it was set up to basically assess the adequacy of current U.S. rules, regulations, and institutional arrangements and processes to prevent the destructive application of biotechnology research and recommend new practices and changes that would help us going forward. The report that was produced, published first in October 2003, was called Biotechnology Research in an Age of Terrorism. Here are the committee members. You'll recognize a lot of your colleagues. And you'll see that it's a mixture of distinguished scientists and people deeply embedded in the security world as well. Here's the recommendation. There was a whole set of recommendations, but the fundamental conclusion of this group is that at some point in the future, we're going to have to come to the day that we came to recently with regard to the recommendation of redaction. They could not imagine there would never be a situation like this, and I can't imagine either. And they recommended that we needed a mechanism in the government to deal with making such decisions, and that was the establishment of the NSABB, the organization that Paul Keim is now chairing. Because the chair of this committee was Jerry Fink at MIT, it's known as the Fink Report. Its actual title is a mouthful. And here's the recommendation for formation of this group. And I want to emphasize the fact that they pointed out that, quote, because of its important bridging functions, its members should include both leading scientists and national security experts. That is, they recognize that this is a decision not to be made by scientists alone. So I was involved as president of the Academy in a very controversial issue before the NSABB was there. This was a paper that we eventually published in July 2005. It was about botulinum toxin and how terrorists might contaminate the U.S. milk supply. And I was approached by many people in government, urged not to publish it. But I tried to see if the NSABB would consider this, but they hadn't met yet, so I had to make the decision. And of course, the default decision is always to publish as was stated, because all the reasons that I point out in this editorial, science advances and protects us in unknown ways, and providing the information only to a select group of people. You can't really guess all the people who could contribute to making progress. So here's the manuscript that we got in August 2011, the one that Ron Fauci was describing. Here's what happened. We sent out the referees for review. This is our normal process. The technical review was very positive, and of course referees always have suggestions for improvement. So in third week in September, the manuscript was sent back to the authors for revision. We got the revision back. We recognized from the very beginning that we would have to do a separate review of technical issues and security issues, and this has to involve different people. The people who do the security review, scientists, but they have some experience with these issues. Most scientists have no such experience. And so we began this review at the very end of September. Then a week later, we got an alert. I got a phone call from the U.S. government saying that this was going to the NSABB and we should wait for I agreed that we would wait for their judgment. Meanwhile, we finished the technical review of the paper, and so as far as we were concerned, if there were no restrictions, this was ready to publish. Then at the very end of November, we were notified of the NSABB decision and recommendation to publish in redacted format, and we've been dealing with this ever since. My assistant has tabulated more than 25 conference calls since then with authors, science, and Nature, and I want to emphasize that the editor-in-chief of Nature, Phil Campbell, and I have been collaborating closely, and we're trying to do exactly the same thing. Our initial instinct was to support the NSABB recommendation because we can't imagine a better system for making these decisions than the one that has been established that closely involves scientists. However, to our surprise, in the intervening years since the NSABB has existed and we've known about this potential day when there would be a reckoning that something should be redacted, there has not been a mechanism developed to assure that those with a need to know, those scientists with a need to know the missing information in a manuscript like this can get it. And so in my public statement made at the beginning of December, I said, and this is still the case, that our response will be heavily dependent upon further steps taken by the NSABB and or the U.S. government or the WHO to set forth a written, transparent plan to ensure that any information that is submitted from the publication will be provided to all those responsible scientists who request it as part of their legitimate efforts to improve public health and safety. So the future is still unclear. That's a matter for discussion. As you just heard, the NSABB will be meeting again to consider some new data, and as I've implied, given my history especially, it's my intention to try to comply with the NSABB recommendation, providing we can have a mechanism for selective access to the redacted information by those with a need to know. Thank you.
Thanks, everybody. I would just add that in this entire process, there's been a lot of voluntary and responsible actions by individuals. As you've heard already, the moratorium, the delay in publication by the manuscripts was all a voluntary type action by the authors, the investigators, and the scientific journals. Questions, please come to the microphone. And when you ask your question, I would appreciate it if you would state your name, please.
David Malakoff, Science Magazine. Tony Fauci, I would just want to be clear. So you're asking NSABB to reconvene to look at revised manuscripts from both research groups.
Correct.
And do I understand that one of the key elements that happened at the WHO meeting is that there was the presentation, further clarification of old data, but also presentation of new data related to both manuscripts?
Correct.
Can you characterize how that new data influenced how people viewed those papers?
That would be difficult because it was a combination of old data that's clarified and new data that's juxtaposed with the old data. I would love to tell you, but the deliberations that took place in Geneva at the WHO were under a combination confidentiality agreement and something that is called export control, which means that when something goes and is discussed or given overseas that has been restricted in its information, there are laws that prevent you from talking about them. They're very complicated. I don't want to waste anybody's time trying to explain that. But the answer is yes, there was new data. Yes, there was significant amount of time for cross-discussion to clarify issues. And if you listened to Ron Foucher's talk, which I'm sure you did really carefully, there were things that he emphasized that he wanted to make sure was clear, because at least certainly in the press they seem to have been unclear.
And question for Paul Keim and Mike Osterholm. Do you anticipate that this new review will lead to a different conclusion?
We can't say anything to that yet. I mean, this decision to come back to NSABB is so recent. Anticipating what the board will do in a short period of time is just impossible. Mike?
Yeah.
No.
Actually, can I just go a little bit further? There was also a strong feeling in our decision to reconvene the NSABB that we wanted to give the NSABB the benefit of having the same information, the same discussion that took place in Geneva. And we wanted to make sure that was the case. So that was the reason, to make sure that you have a balanced, fair situation where you don't have one group hear one thing and not others, that everybody hears the same thing, because there was obviously a disagreement and recommendation between the NSABB and the Geneva group, and we wanted to make sure that all the data and all the discussion is balanced between both.
Next question.
Roxanne Kamsey with the news section of Nature Medicine, and this question is for everyone. I was curious to know whether there are any security measures being taken to protect the data right now, because we live in an information age. I wonder if there are people who aren't waiting for a publication but might be trying to access the data as it's emailed or discussed on the phone or anything like that.
Ron, do you want to? I mean, one of the problems with security is you don't discuss it in an open forum, but maybe you can be reassuring at least.
That's right. And so one of the problems is, of course, that scientists are usually forced to be open about everything they do, because we're working with taxpayers' money. And so did we do that this time. But certainly there have been cleaning operations, so to speak, to get our computer systems cleaned up. ICT security, as you all know, is just not doable. So any good specialist could hack different systems. The manuscript has been shared with NSABB members, journals, and amongst the authors. We're doing everything we can to protect the manuscript. We already did everything we can to protect the virus from getting out of our facilities. And I will leave it with that for now.
Thank you.
Bill Schultz, Chemical and Engineering News. Dr. Foucher, I hope I'm not misunderstanding, but you seem to be saying that the virus you created is not particularly lethal in the ferrets, and yet, Dr. Keim, you have said to me in a phone interview that this was, quote, the doomsday virus. How are you coming to such different conclusions, and what is the public supposed to understand?
So I'm sure I didn't actually say that, but I have heard people call it that. I did not call it the doomsday virus, I should say. Ron?
Well, so the virus is highly pathogenic in chickens. The virus is pathogenic in ferrets if you put a lot of virus straight down in the lower airways. But it's well recognized by everybody in the flu field that if you take H5N1 and stick it up the nose of a ferret, the ferret really only gets the flu, a normal flu. Now, our virus is slightly different. It causes some neurovirulence, but certainly if the ferrets receive virus via aerosol route, we have never seen severe disease in the ferrets. Now, a ferret is not a human. And as Mike just pointed out, there's still serious debates about how fatal this virus is in humans. I think more research is needed. I do not think that the 60% case fatality rate is correct. What it is exactly, we don't know, and we need to do more research. But certainly, this would not be a virus that would kill half of the world population, as we've seen in the lay press time and time again. That is clearly wrong.
Yes.
Just an important point to add here is that there's been much of the emphasis placed on virulence and the ability to kill. And as has been discussed by Ron and others, and we know that the extrapolation from a ferret model to a human to what the virus might do if it were circulated in humans is very different. The key piece of information here that seems to be missed often, though, is that one of the very classic experiments of concern that the Fink report emphasized, in which we took great consideration, is changing the host specificity of a virus capability for transmission. And that is different than talking about virulence, and yet that's a very critical piece. So just merely being able to make a virus with mammalian to mammalian transmission, that's a very important issue. And I don't want to say that that should be the deciding factor either, but that's a critical piece.
I guess I would add that there's great uncertainty here. I mean, especially now with the moratorium, Iran isn't doing any more experiments in this area. And so exactly what all these parameters are not as well defined as perhaps is necessary to make an ultimate decision. It was in this environment of uncertainty that the NSABB came back with its recommendations to not fully publish. And again, I think the idea is it's time to have a pause for us to get a broad discussion going globally and across different segments of our society. And in fact, the recommendations from NSABB clearly can be changed in the future. But what we also recognized is full publication right at that time could not be reversed. Once the papers were out, they were out. We can go back and reverse this if that's the best course of action.
So to just clarify a bit, without predicting anything, without talking about any processes that will unfold, to the perception of, I think, most of us in the room, there was a perception, however it got there, that, and we've seen it on television, I've seen schematic diagrams of it on television, of a ferret in one cage sneezing on a ferret in the other cage. That ferret sneezes and they all die. This is what Ron has just said. I didn't say it. Ron just presented that. That is not what happens. When a ferret sneezes on another ferret, the ferret gets infected, gets flu-like syndrome, doesn't get sick, recovers, and does not die. And I think when you talk about forgetting about any of the process, just the perception that's out there. Am I correct, Ron?
Absolutely, Tony. These viruses do not kill ferrets if they are sneezed upon. And the second point I was trying to make is that it is even unlikely that if this virus would be released out in a ferret colony, it would spread like wildfire in ferrets. And so to then extrapolate that this virus would spread like wildfire in humans is really, really far-fetched at this stage. You know, if anything, our data suggests that this virus spreads poorly. It is aerosol transmitted and therefore scientifically very, very interesting to study. But whether this virus would, if it would ever escape from a lab, would start spreading, we don't know.
Next question.
Hi, Daniel Gillet, French CBRN program. I was wondering if NSABB expresses recommendation about whether destroying the strains after the study, or if not, who does this?
I didn't hear him.
We can't hear you.
Yeah, I'm sorry.
Strain destruction at the end of the study.
Strain destruction.
The NSABB did not make any recommendations as per that, and I'm sure we had discussions, but it never reached the point where we were making recommendations for strain destruction. So we didn't deal with that issue. A lot of things we didn't do.
I'm Raymond Zelinskas from Monterey Institute. I'm not a journalist, so... but I do.
Can you speak up? We're having a really hard time hearing up there. I'm sorry.
Okay, I'm Raymond Zelinskas from Monterey Institute. I'm not a journalist, so I might, I guess, approach a little different. So I have two questions, both to Dr. Osterholm and Dr. Keim. The first one has to do, why didn't you have an NSABB-like process at the beginning of the process when you were actually considering, when the NIH was considering the proposal? And instead of coming at the end and saying, oh, we have a problem, we got to deal with it. The second has to do with, it seems to me this research is really basic research. And the possibility of applications coming out of this is very, very distant, whether it's vaccines, improved vaccines, or diagnostics. And it would seem to be the same thing for using it for ill purposes. So even if you have, as Dr. Osterholm suggested, that you can get mutants for a couple of $1,000s in a couple of months after it's published, that doesn't mean that you can actually use it for anything. So the NSABB process really has to do with dual use, but maybe that's the wrong approach. It should be really have to do with the risk issue, which is more immediate, having to do with the risks of the organism either escaping or being transported out of the lab.
Mike, let me answer the first question. I'll let you answer the second one on risk-benefit analysis. So something that needs to be clearly explained is the NSABB is an advisory board, and we only do what we're asked to do by the U.S. government. So we're not like the GAO. We're not out snooping around looking for problems. So the reason we didn't review these projects when they were first being funded is because we weren't asked. And so that's the simplest answer for that is the NSABB does not go out and do this. Primarily, we've been formulating policy recommendations, and we have whole stacks of those raised, which you're welcome to read. But we weren't asked, and so we're not a part of that part of the process. Maybe we should be, but we weren't in previous times. Mike?
Yeah, let me just follow up on that. We agree that if we ever have another experience again like we just had, where we got in at the end of the process, that's a failure. This whole system needs to be reevaluated for how that early involvement that you talked about should be done. So I think what I'm finding is we have violent agreement among most of the life sciences community that that's where it should be. So I think that's number one. Number two is, I just want to clarify that when we looked at the issue of countermeasures, as you raised about vaccines and so forth, we have to be really honest with ourselves about what our countermeasure armamentarium is and how well it works. We just went through the third pandemic where vaccines were available. And for the third time, the vaccine community failed to have any substantial impact at all in the pandemic. The reason for that was that the vaccines arrived too little, too late. And in our country, less than 16 million doses were delivered to a population of 300 million people by the time the second wave was over. Worldwide, even the same situation occurred in Europe as well as in other developed countries. In the developing world countries, the WHO, after a year, had only delivered 78 million doses to 77 countries. So the point being is we need better countermeasures, ones that are more effective and more timely. These data, whether available or not, do not fundamentally affect that. And so our whole point has been is that we need a much better influenza system. We can no longer accept the vaccines that are here today as being sufficient. The same is true with antivirals. I could go through that and what happened there. As far as surveillance is a very critical issue. Again, I think we'd have violent agreement on that. But the problem is the global surveillance system for H5N1 is seriously broken. It is for influenza viruses in general. Now, we believe that has to be fixed. But merely changing the color or the cover of a smoke alarm that doesn't work because it has no batteries or electrical source doesn't fundamentally change it. And so we're saying making these mutational data available doesn't change surveillance. And I don't want this to be misinterpreted. We don't think surveillance is important, it should be changed. We really do. But last year, less than 160 sequences, not even full viruses for H5N1, were put into GenBank. You can say that was not all of them. But the bottom line is we have a globally inadequate system for surveillance. So making these data available on a general basis would not materially affect the overall global surveillance for H5N1. At the point where someday that might happen, then we could see a very different possibility. Finally, as we all know, these are not the only mutational changes. It could be a prediction of a pandemic strain emerging. So while it still is helpful, we wouldn't want people to walk away saying if you just had this cassette, you would know that in fact this is the new pandemic strain about to emerge.
Can I go back to the audience, or Tony?
No, we don't have enough time to discuss adequately all of the implications, except to say that I certainly agree with Mike that we need to better our capability of responding with vaccines. That's an issue. That's a lot of a technical issue, a capacity issue. Mike has spoken eloquently about that for years, and we have addressed that in our pandemic preparedness plan. But I just want to make sure to understand that understanding adaptability and transmissibility, which is what the NIH and all our advisors have recommended strongly that we do, understanding how a virus adapts to a host, how it transmits and readapts to another host is a very, very important problem in understanding influenza. So I think that what we need to make sure is that we don't mix apples and oranges, is that we have a problem in that we need to be able to respond better with vaccines. The federal government has invested billions of dollars in trying to convert a very antiquated vaccine development program into a 21st century program by partnering with the industry. That's important, that's real, we totally agree. On the other hand, from a scientific standpoint, it is really critical to understand the biology, the virology, the molecular epidemiology of how viruses go from one species to the other. Because if you look historically throughout pandemics, that's how it happens. So I just want to, that's the rationale for why these studies are done.
Thanks, Tony. Next.
Heinz Hellerbrock, Robert Koch Institute, Berlin, Germany. I have a more general remark. In the keynote lecture on Sunday, Paul Offit had a remarkable point to me. He said, although scientists get it wrong all the time, science is enormously self-correcting. And I thought he was talking about the scientific community in its entity. I'm not sure if this task should really be taken over by a board of 20, 23 experts or so. I think this should be really left to the entire community to discuss, because we are discussing points here. We are discussing mutants nobody knows about. It's a little bit weird to me. So just as a general remark.
Yeah, so the board agrees with you. We think the scientific community should be engaged in this issue, and the scientific community, policymakers, and the public should be engaged in this issue in making decisions, not just 23 people. Mike?
Yeah, and let me just follow up. I'm sorry I couldn't hear all of your question, but I'm speaking personally now, not on behalf of the NSABB. But I am one of those influenza researchers that has been very supportive of the work that Ron is doing and thinks that that type of research, as Tony just laid out, is very critical research. Our point has been how it can be done safely. This has been a misinterpreted comment, but I really believe that of all the agents that could accidentally leak out of a laboratory, for example, there is none like influenza. Influenza is the equivalent of having a screen door on your submarine. Once it's out, it's gone. Smallpox, any SARS, any number of viruses, it would be a horrible tragedy if they ever were to leak out, but we could contain them and it would not see a global pandemic. So I think that, again, issues around safety, issues around who has access to the virus. I've not personally been worried about somebody in a cave somewhere, which has been characterized often as the terrorist approach, is the one that's going to do this. I worry about the garage scientist, about the do-your-own scientist, about the person who just wants to try to see if they could do it. And today you may say that's difficult, but in fact it's quite remarkable to see what's happening out there with sequencing, et cetera, and so forth. And so I just point out that I think all of us really have at the heart, we need to know much more about this. The real risk we have today is Mother Nature-made pandemics. But there are legitimate concerns to make sure that we don't have a problem. 1977, H5N1 was an accidental release, no doubt about it. We don't want to see that happen again with a virus like this. What we're trying to do is come to the middle. How to do this research safely, how to do it in a way that furthers all the points we've just talked about, and at the same time, make sure that we don't create our own problems here. And that's why I think we're asking for this pause to think about this and to move forward in a positive, proactive way.
So one last question, Petra.
Well, I suppose it's... Can you hear me? I'm too short for the microphone. I was just thinking, I've been listening to this, and actually I feel that this is quite a positive situation. The peer review system worked, it caught the papers, there was a system came into play that allowed people to discuss it. So personally, I feel this is a quite positive thing. Okay. I think there's a bit of concern about how we then identify who are legitimate researchers who can access the data in order to further the needs of mankind. But I think what this and so many other cases show is actually journalists in the common press have an awful lot of responsibility here. It's easy for them to knock the scientists doing this work, and it's easy for them to use terms like doomsday bug. It's lazy, sloppy reporting. And I think you should be bringing just as much effort to bear on making the journalists behave responsibly as you do to making the scientists behave responsibly. So.
So I'm going to ask the U.S. government to send that issue to NSABB.
Thank you.
Now with that, join me in thanking the panelists for what was an excellent session.